First of all, thanks for having me. It's an honor to give a lecture in the C-STAR series where so many great researchers who inspire me have given lectures. And today we'll be talking about the neural basis of sentence structure complexity during speech production. And that's just a very, very long title, mostly to summarize the two resources that we'll be using to basically talk about how we try to explore the way the brain processes syntax and sentence structuring as we produce speech. So we'll be talking about natural language processing and lesion mapping. I have no disclosures to report for this talk, and these are some of the goals that I will try to achieve during this 45-minute talk, and then hopefully we'll have some time for questions. But basically, I want to make sure that we're able to highlight applications of natural language processing and how we can use that for aphasia. Basically, try to explore the way we can use lesion-symptom mapping to study the neurobiology of language and understand how we can infer information about the neural basis of syntax as we process speech. And discuss very exciting new lines of research that open up based on these preliminary data. So basically, the motivation for this study comes from the idea that even though much information can be transferred or communicated through isolated words, you still need this complex communication that requires a sort of a macro or a supra-organization of all of these little tiny pieces of information, which are words, and organizing these elements within a structure that is shared both by me as the speaker and you as the receiver, and that we both understand those same structures, make us able to communicate more symbolic information or more complex information. And if you think about it, as we produce speech, this is all about stringing together different pieces of information, and some of these are have objective meaning, like just mentioning a chair, naming chair. And then there are words that we use that have a role or a practical meaning in that structure. For example, the word for. Now, if you think about these two sentences, which I really like, which both have the same words, it's just that the order is different. You can see how the first sentence, with a polite attitude, the professor proceeded to help the elderly man in the cafeteria, is somewhat different to the second sentence, which again has the same words. In the cafeteria, the professor proceeded to help the elderly man with a polite attitude. So much so that even the second sentence can be ambiguous, right? Who is being polite? Is it the elderly man being helped by the professor, or is the professor helping him with a polite attitude? And so this just shows us how even during everyday, regular, non-aphasic speech, we sometimes have these subtleties that clearly show that language, and I'm assuming everyone who's watching this conference right now is passionate about language and finds it incredibly fascinating as a function, but part of this fascination comes from how complex it can be. And syntax and basically structuring of these sentences basically has a strong role in making this such a challenging endeavor. And so if you think about the different aspects of language that have been studied in the literature, most of them have been studied through the use of constrained tasks. And by constrained tasks or focused tasks, we're talking about things like picture naming, sentence completion, all of those tasks that we use in the laboratory and the clinical settings that give us very valuable information. I think it's undeniable how much we can learn from those tasks. But when you think about the daily demands, the demands of everyday living, and you think about how speech is produced, the truth is that some of these tasks, these focus tasks, fail to capture how much of the day-to-day communication has all of these challenges that we're talking about, right? With the complexity of syntax and sentence structuring. So they sort of fail to capture that day-to-day communication and basically limit this ecological validity. You know, are we truly measuring, for example, speech production if we're just using a task that is asking the patient to give us the last word of the sentence that would make sense in that context? And so that is a really interesting challenge from a procedures perspective, from a methods perspective, because we are basically unable to capture some of these ecological or more subtle deficits sometimes. So what is the alternative, you may ask? Well, we can produce, or we can challenge patients with tests that are more capable of eliciting more day-to-day or real-life demands in terms of communication. And so there are a number of tasks. These are just some of the examples: picture description tasks, storytelling tasks, and semi-structured interviews that may give us more connected speech. And that is basically the key of these alternative tasks. The idea that you're producing speech that flows in a way that mimics everyday scenarios more naturally. Now, with this alternative comes a very important cost, which is that for you to analyze connected speech, you require a level of training and an amount of time that most people don't have, or that in groups tend to be confined to research. And for example, from a clinical perspective, if I take my researcher hat and put my clinician hat on, it's very challenging and very demanding and very costly to have someone analyzing the connected speech elicited by patients with these tasks. So it is really interesting to think about how we can go about trying to overcome these costs that come with the alternative of connected speech analysis. And one of the potential applications of what I'm going to show today is perhaps this one. And I'm going to try to convince you throughout this talk that even though these are first preliminary steps, we're in a relatively good place to start using some novel computational methods. And one of those methods is natural language processing. So for those of you who haven't heard about NLP, it's at the intersection between linguistics and computer science. And basically, it's applying automated computational algorithms to analyze human language in a way that is automatic. This is not an automatic processing of speech. This is not transcribing speech into text. This is given text, giving a linguistic human type language input. Can you automatically analyze it to extract different features? And I'm going to be showing you some of these features today. But if you haven't noticed, NLP is already being used widespread beyond the area of research or research in aphasia, as I'm going to show you. Biomedically, it's being used to extract data from massive amounts of clinical charts in hospitals, not only for billing, but also to try and understand quality improvement measures, etc. Recognition systems of speech actually use this. Siri, or what is it, Google's Alexa or Amazon, they're all using NLP. And search engines, for example, use NLP to maximize or to optimize their searches. Now, NLP in human research has been somewhat limited. These are some of the examples in which NLP has been used. So they've used natural language processing to analyze grammatical errors produced by speakers that were learning different languages. It's been used to identify individuals with MCI, trying to determine if there are any precursors of the neurodegenerative disease that may come from language and from more subtle things than a Boston naming task, for example, where these patients tend to perform at the ceiling level. It's been used to classify subtypes of primary progressive aphasia based on lexical and syntactic profiles, so trying to determine different subtypes of PPA, like semantic dementia or progressive non-fluid aphasia, based on the speech elicited by these patients. And more recently, it's been used to classify different kinds of paraphasic errors in a semi-automatic manner. However, it's been not used to the best of our knowledge to investigate how patients who have aphasia or some degree of aphasia following a stroke of their dominant hemisphere, how are they producing speech? Are they having subtleties, or are they having patterns of speech production deficits that are not captured by constrained or focused tasks? And to us, this was an important point because so much of agrammatism, and there's a great talk by Dirk Denote in the C-STAR website about agrammatism, but if you review it, you'll see that the hallmark of patients with aphasia is characterized not only by a lot of variety in terms of their agrammatism, but also by some things that happen over and over and that have to do with this deficit in morphosyntactic encoding, the fact that they tend to favor simple utterances, very short and simple utterances. They tend to lack subordination, so they tend to miss words or omit words that help subordinate different clauses, or they tend to omit or substitute different function words. And so these are some things that are hard to obtain when you're just analyzing patients either with a brief battery for language or with some of these focused tasks. But these things definitely come through when you start talking to the patient, especially at bedside or in the clinic, and you notice that they have this effortful speech a lot of times at the expense of being unable to construct complex sentences to deliver the message they want to deliver. And so with this idea in mind, we said, can we try and map in the brain where this deficit in performance in terms of sentence structuring is coming from? And lesion mapping is a great way to go about this, because contrary to what functional studies do, this is a way to actually determine what areas are critical in the brain for the performance of a behavior. We've learned a whole lot from functional studies, but we cannot assume that whatever activation we're seeing, for example, in fMRI, is necessarily a structure that is critical for this activation. So you may be well aware of this classic dissociation where if I injure or if I have an injury in area X and I can no longer produce certain function A, but I can still perform B, but an injury to an area Y has or is associated with a deficit in B, but not in A, then you can say, well, X is probably critical for A, but not for B, and vice versa. And so this is the basis for lesion mapping. And you can go about lesion mapping in maybe at least two worlds. I like thinking about lesion mapping in the gray matter world, where voxel-based lesion symptom mapping helps us understand this question, right? How does damaged tissue relate to behavioral deficits, looking at a voxel by voxel approach? And I believe there's also a lecture somewhere in the C-STAR website that introduces lesion mapping. This is the classic Bates paper from 2003, where they introduced the idea that you can, at each voxel, try to determine, based on a group comparison between those that have and do not have a lesion in that voxel, are their scores, are their behavioral scores different? So this had a lot of advantages, right? We didn't no longer need to determine what is a behavioral performance deficit, so no longer needed to determine, you know, under this score, I'm going to say this person's impaired, above this score, this person's spared, and that behavior. You didn't need to define specific regions to say, is this lesion or not? So much of lesion mapping in the early part of the history of neuroscience gave us valuable information, but it was based on a macro assessment of, well, it seems like Broca's area is damaged, so they're not doing this, but you know, there are many things that come with big lesions that give you subtleties that at the voxel by voxel basis, you can learn so much from. And so the gray matter world, we can approach lesion symptom mapping with this voxel-based lesion symptom mapping. I'm going to refer to that as VLSM. And then from a wide matter world, there's a great talk by Dr. Bonilla in the C-STAR website as well about how we can use the connectome for aphasia. But we can think about using the connectome in terms of not just understanding the way it can predict things about aphasia performance, but also thinking about functions per se. So just like voxel-based lesion symptom mapping looks at what gray matter structures are damaged with an associated behavioral deficit, well, can we think about what wide matter tracts, when damaged, are also showing us that they're critical for the performance of a given behavior? And that's basically what I'm going to refer to as CLSM, or connectome-based lesion symptom mapping. We recently published sort of a summary of the method behind this and how it's been used so far in the field of cognitive neuroscience and language in particular. And it's been recently published, so it's a little outside the scope of today's talk to go over the details of this, but I think if you refer to this paper and Dr. Bonilla's talk, where he shows how to build a connectome, you'll have a good understanding of how this can really open our gray matter sort of focus and really look at the brain from a different approach, which in aphasia is so crucial, because basically the diaskesis hypothesis and the connections are changing even when gray matter is spared. So for us, it was really important to look at both worlds, the gray matter world and the white matter world. And we did this again with VLSM for gray matter and CLSM for white matter. This is just to show you an example of what I was saying. It can help us understand how language is processed in the brain. We published this a few years ago showing how the production of nouns, or really the production of words that are able to name objects, versus the production of words that are able to name actions, actually depended on two dissociable, very clearly distinct networks. I think for the online people, I have to use my cursor. So hopefully you're seeing, as I move my cursor here, that there's a frontal network, more dorsal, that involves frontal structures that supported the production of action naming, the naming of verbs, whereas we showed that connections that involved structures in a more posterior ventral network supported the production of nouns. So this is just to show you one of many examples out there of how connectome can be used to understand language in general, not just in disease processes, but using aphasia in this case, and in the case that I'm going to show you today in our research, really as an excuse to understand language in general, and also gain understanding that can, in the same process, help clinically, but with really interesting theoretical and clinical implications. Now, the good news is that lesion mapping is pretty accessible, and it's easy for me to say because we have someone like Chris Riordan and everyone in the team working on developing these very friendly algorithms that, just by inputting some basic information, produce really interesting and really powerful computational calculations and computations. And if you want to know more about NISTAD, which is the package that we use developed by Chris and his team, that's their website. And this is really a friendly, very easy to access method to go about lesion mapping. So if we jump straight into the data that I want to share with you today, this is a study that was done based on the ongoing cohort of aphasia patients at USC and MUSC and all the C-STAR consortium. And these are 65 participants that had a unique stroke in their dominant hemisphere. And we used chronic patients with stroke, so they had to have at least 6 months since their stroke. They had no history of other neurological disorders, so we ended up excluding everyone who had, for example, a hint of dementia that was concomitant, or other disorders that could mask behavioral performance. Almost 2/3 of our participants were male. You see their mean age there, about 58.5 years on average, and they were evaluated a mean of 37.2 months since their stroke, so truly chronic stroke patients. And not all of them had aphasia, so you'll see that there were different patterns of aphasia, and this was very favorable for our study because we really wanted an array of behavioral performance, because the more diversity you have, the better, or I guess the more reliable it will be to determine whether a specific region in the brain, whereas gray matter or a white matter tract with lesion mapping actually maps to specific behavior. This is an overlay of all of their, an overlap of all of their lesions. So you can see that the closer you get to red, the more overlap between patients. And so you'll see that it really, at the group level, is a cohort of patients that had lesions involving the perisylvian fissure, the perisylvian structures, that of course is more likely to involve language. So I started my talk by focusing on how we've learned a lot from constrained tasks, but they failed to capture everyday challenges and everyday scenarios in terms of language. And so for our research studies today, we used a combo of 3 tasks that are all picture description tasks. They're all widely used in terms of clinical use. One is the very well-known cookie theft scene from the Boston Diagnostic Assessment Examination. This is a very well known to clinicians who are not researchers. It's part of the NIH stroke scale. So anyone who's been trained in neurology or SLP probably is aware or has seen this, if not used it. And it's a task that tends to elicit a lot of different words and actions and objects. We use the picnic task from the WAB assessment battery. And we use the circuits task from the Apraxia battery for adults, the second version. And so what we did is we challenged patients with these 3 tasks. We allowed them to describe the pictures for 2 minutes each. And we used other measures of language because, as I will show you later, part of the challenge was really trying to exquisitely try to map sentence structuring. But when you think about everyday speech and when you think about a task like a picture description task, there's so many confounding variables, right? The ability to name, for example, if I'm going to name an object, I need to show that my ability to build a sentence is an ability beyond my capacity to just mention, yeah, this is cell phone. But for me to say, I am calling you on my cell phone, that has to be a different level of processing, or so we hypothesized. So we use the WAB battery really as a measure of global aphasia severity. And you'll see that I'll show you, we also use the naming subscore of the Westman aphasia battery, which is made up of 4 different subtasks, each one of them associated with a number of points. And the total is then sort of transformed into a total naming subscore. And we focused as potential confounders for our behavioral main task, which was the picture description task, on these 2 variables, naming subscore, the total one, but also we looked at sentence completion, mostly because we were interested in seeing if patients' ability to elicit a correct name in the context of a sentence was spared or impaired, or what was their performance on that task. And that's what the naming subscore sentence completion task does. It gives the patient a task, and then the last word is missing, and so they need to complete the sentence with the appropriate word in the appropriate context. Now, if we think about aphasia and how speech production can be impaired, apraxia of speech is another really important confounder. And so we use the apraxia of speech rating scale, which is this 16-item 5-point scale rated by the interviewer based on a number of things. It includes the picture description task from the WAB, and it's a task that has some subjective component in that it is a human judging another human based on operationalized criteria, but there's definitely always room for variability. But it's a very, very well-used task as a popular task for somehow trying to quantify a challenging thing to quantify, which is apraxia of speech. So this was another task that we used as a potential confounder. So I've told you about our main task. We showed them 3 pictures and asked them to describe each picture for 2 minutes. And I'm going to try and take you briefly through the process that we took to derive the variables that we ended up trying to map to the lesions. So the very first step, and this is not a step that is included as part of natural language processing. This is a step that involves still a human trying to interpret what the patient said. But the first step was trying to verbatim transcribe the speech elicited during the tasks. So we had human transcribers writing word by word what they heard, and this included fillers. So that's why I'm giving you this example here. So if the patient said the boy is stealing cookies from the jar, those fillers were there. If there were paraphasias, they were there to the best of our possibility. You know, I'm sure if I said kakis instead of cookies, you could argue, do I type that with a C or with a K at the beginning? It really didn't matter for our overall syntax analysis. So the transcriptions were done verbatim to the best of the possibilities. Then a second step, which removed the fillers so that we would have them the sentences without fillers, because when you're analyzing syntax and you're going to do this with a machine that does it for you, it can be very tricky to have the machine identify the filler and what comes after or before and what role it plays in the sentence. And the third step, which was a very challenging one, so we had to somehow define with a very clear threshold, and I'll tell you about that in a second, was based on the audio of the transcriptions, or of the performance of the patient live, we introduced periods and commas. So a comma was anything that was a short pause, and we defined this arbitrarily as less than 2 seconds. Other studies had done this, and so we followed that rule. Less than 2 seconds was entered into the transcription as a comma. And then periods were used for anything that was longer than 2 seconds, or, as other studies have done before, whenever the person working with the transcription felt that there was a sentence boundary based on a finished or completed idea or a finished clause. So if the patient had changed to a completely different aspect of the picture, then it was fair to give that a full stop and make that a sentence. And so if I go back, we went from the boy is stealing cookies from the jar to this phrase at the bottom, the boy is stealing cookies from the jar, period. And if you think about this, this was done for 6 minutes of speech. So it's definitely a lot of valuable information that we get there. This is not just one sentence, but 6 minutes of more like everyday real scenario connected speech. So, and I'm sure these are some of the concerns that are being raised in your brains right now as you hear this task. And so we try to address some of these concerns early on before going through with the variables. So one of them was inter-rater reliability. You know, what I call a comma, you may call a period. And how do we know that this is a fair assessment of what you're hearing in the recordings of the descriptions of the pictures? So the first inter-rater reliability concern we addressed by looking at a subsample. I think we did 15, where 2 different independent blind reviewers, so to speak, or raters, did their periods and commas in the transcriptions, and then we used intraclass correlation coefficients to determine with a 2-way random model whether there was a decent intraclass correlation. And the truth is that there was. Clearly, the 2-second pause threshold helped, and you'll see that for the commas there was a really high 0.98 intraclass correlation, and for periods 0.93. And probably periods are a little more challenging because of this idea that you can arbitrarily say, well, there's a period here because it's a completed idea. And especially in the patient with aphasia, if they're saying stealing window, well, are they talking about the same thing or not? So even with that difference between commas and periods, 0.93 intraclass correlation is pretty impressive and pretty reliable for behavioral research. A second concern that we had was, well, is the parser valid? And I'll show you what I mean by that soon, but the first step is understanding what we did with these transcriptions. So once we got the verbatim transcriptions and they had their commas and their periods and they were analyzed for the basic structure of what the patient was trying to say, then we moved on to the automatic step, and that was the natural language processing. So from the text input, we used the Stanford parser, which basically identifies parts of speech and tries to assign each word in the sample a tag. Is this a noun? Is this a verb? Is this noun part of a noun phrase? Is this noun part, or is this noun phrase part of a clause? And is this clause part of a sentence? And I'll show you examples of this soon. But that's basically what the parser is doing, is identifying parts of speech. And then there are a number of syntax or syntactic complexity variables that can be derived. This researcher and his group, Lou, had this online process where, again, you input the text, it goes through the parser, which uses the same parser, the Stanford parser, and then it outputs a number of variables that try to reflect on syntax complexity. And I'll show you some of these variables. But overall, for our behavioral variables, we used 20 syntax features that were derived from the use of this parser and the syntactic complexity analyzer that Lou has online. So that's from the data gathering or the data processing perspective. From a visual perspective, we use this natural language toolkit to transform the output of the parser, which can be pretty difficult to just codify by just looking at the output. And so looking at trees, the way we all learn to dissect sentences in terms of grammar, is a lot easier. And so these are the features that we derived. We had things that variables that really just measured more of a quantity aspect of the sentence. So things like sentences, number of clauses, number of noun phrases, and number of verb phrases. But then we had other features that really said more about the syntax complexity of the speech produced. So things like the height of the clause, and I'll show you what we mean by height and by width that you'll see here. The idea that you can have a distance between noun phrase to noun phrase and noun phrase to verb phrase, and so forth. And so this is what the trees look like. You'll see that this is a very simple sentence. The young boy is stealing cookies. And you'll see that the parser determines that this is a sentence that has a noun phrase and a verb phrase. And that noun phrase has a main noun, the boy, with its determiners and an adjective that precedes it. And so in terms of our variables, when we talk about width, we're talking about how many elements are there inside, for example, in this case, a noun phrase. So how many leaves are there that branch off this one element. And then you'll see that we also have a measure of complexity that is height. And so that's how many of these branches are within one element. And you can see from the second example that the more complex the sentence becomes, of course, the more complex the analysis is. And this is where the NLP comes really handy, right? Imagine doing this as a human being on 6 minutes worth of speech for 65 patients. That's a real challenge. And that's why we think NLP can be such a potential powerful tool for the analysis of aphasia speech. So the third concern that we had is, well, we showed that, sorry, I haven't talked to you fully about the second concern. This is the one. And is it valid? You know, what the analysis that is being done by NLP, is that enough to say, yeah, I trust what this is doing. This seems to be doing things right. And so one way to go about it was validating against healthy controls. So we presented 10 age and gender matched controls, the same picture description tasks. And what we did is we compared the output of these 20 syntax variables derived from NLP with the output of those same variables produced by those 19 patients that had the stroke on the left hemisphere, but did not have aphasia by the time we assessed them. This is based on the WAB-AQ. So when I showed you the different language variables, I told you aphasia severity was measured with WAB-AQ, the aphasia quotient. So these patients didn't have aphasia, but they had a left hemisphere stroke. And so you'll see that after correcting for Bonferroni, there are no significant differences for any of the 20 syntax features that we had derived. And so, oops, I'm sorry about that. So for us, this was important because at least whatever output we were getting for these patients that did not have aphasia was comparable to the output that we were getting for healthy controls who had not even had a stroke. Now, a third concern is, all right, they seem to compare, or their output is comparable to that of non-stroke patient or no-stroke participants. How does the parser perform independently in each individual? And so this was a very challenging part of our concern analysis, in that the only way to go about this is to humanly analyze the transcripts or really the output of the parsers and see if we agreed or not with the parser, assuming that our knowledge of how to parse a sentence is reliable. And so with that assumption, we went one by one on each of the sentences, actually each of the words that were tagged, and we said, do we agree with the tagging or not? And we found some mistakes that the parser does. Things like words like fishing, for example, which could be a noun or it could be a verb. And sometimes the parser mistakenly tagged them the way we wouldn't have. And that was based on guessing what the patient was trying to say. So with aphasia, this becomes a lot more challenging. So we sort of classified errors into definitely an error, pure error. If you said that cookie was a verb and it's really a noun, then we classify that as a pure error. And then we had other errors that were less so. For example, if the patient said lion, and so they have 3 or 4 attempts to say the target that ends up being the correct one, the parser does a really good job at classifying that final target lion as a noun, but maybe those first attempts, it'll be like adverb, adjective, noun. And so when we are very, very, very conservative and we take into account all those errors where you're saying, well, the parser doesn't know any better. So if we're very conservative, the error rate was 4.13%. So about 1,400 something words were correctly classified or were incorrectly classified out of over 36,000 total words produced by all the patients in all the transcripts. So I think we can agree that that's a pretty good error rate for an automated algorithm that is parsing aphasic speech. I am pretty sure that if we gave the same task to humans and we asked them to do that for 65 patients in just a matter of an hour, you would get even a higher error rate, right? There's fatigue, there's et cetera. So I was pretty impressed. I was actually expecting a much higher error rate. All right, so taking it all together, we are doing voxel-based lesion-symptom mapping and connectome-based lesion-symptom mapping on these 20 behavioral variables, which are somehow a reflection of syntactic complexity or structure complexity during speech production. Now, it's a lot easier to try and find or sort of reduce this dimension that we're trying to capture, which is really syntax or elements of syntax during speech production. And so what we did is we applied primary component analysis on these 20 structures. And so the first component analysis, principal component analysis that we did was on all 20 features. And then I'll show you why we did a second principal component analysis with 16 features, where the ones that we kept, the 4 that we excluded, were the ones that were more a measure of quantity of speech production. So if I just measure the number of noun phrases and verb phrases, that's not really saying much about the complexity of the structure. It's just saying how much speech was produced. And that doesn't mean that the speech is complex, right? I could just say lion, 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 lion all day, and the parser may tag each one of those as a noun phrase if I'm putting periods in between. And so that doesn't make it complex. And so that's why it was so important to run these 2 different principal component analyses. And I'll show you how we control for confounders, because again, the ability to name, for example, is a crucial ability that that could confound speech production as well as abraxic speech. So these are the results for the first component. You'll see that the first component explained 95.1% of the variable or the variance in the 65 patients. And so it was striking that the factors that loaded more strongly on this principal component were noun phrase, just the total number of noun phrases produced, the total number of verb phrases produced, and the number of clauses. So really what this first principal component was showing us was more of a sentence production component, right? How much speech is being elicited. That's why we did the second principal component analysis, where again, we excluded sentences, clauses, number of noun phrases and number of verb phrases, but we left aspects of the complexity of these. So we were interested in the width of the noun phrase or the height of that noun phrase, right? How many branches were there? How many elements were there in the noun phrase? And so we call this a syntactic complexity component. in that when you look at the higher factor loading, it's all things that really reflect more on structure complexity, like the length of the sentence, the height of the clause, the height of the verb phrase, the distance, so the number of elements that existed between a noun phrase and the following noun phrase. So these are our two main variables that we'll be trying to map, both with voxel-based and connectome-based lesion-symptom mapping. So let me take you through these figures. This first one is a result of voxel-based lesion-symptom mapping, and this is based on regions of interest that involved voxels that were significant. And what you see on the top row is VLSM for that first component of the principal component analysis with all 20 features, so sort of a sentence production. And you can see, right, how the injury to these frontoparietal and maybe posterior superior temporal structures was critical for the production of sentence components. And just to show you an example and show you the reliability of voxel-based lesion-symptom mapping, this is the VLSM for naming performance. So this is clearly a variable that is instead critically associated with the sparing or the integrity of structures in a more posterior ventral distribution, which makes sense when you think about what naming does. And clearly, and this is one of the limitations that I think we need to highlight for our study as it is now, is that because we used WAP naming, there is a bias to action, to object naming. So it excludes action naming. All items in the WAB that are part of the naming task are all objects. None of them are action naming stimuli. Now, these are the variables alone, but what happens when we introduce each other as a confounder? So when we look at this idea of sentence production and we control for naming, you'll see that there's really, and these are the same results. It's just that on the bottom, you're seeing really just the voxels that were significant, where on the top, you're seeing the regions of interest from the atlas that we used that included voxels that were here. So really what you're seeing is this more confined cluster of voxels that involves the inferior frontal gyrus, a portion of the insula and the superior temporal sulcus, when we introduce naming as a confounder for this performance in terms of sentence production. Then there's the second component, or I guess the principal component of the second PCA, which again was based on only the 16 features that were more reflecting syntax complexity than just production or fluency in a way. And you'll see that when we control for WAB naming again, we get this distribution of voxels that are critical for performance of this sort of component extracted from all of these variables, which again came from the natural language processing applied to the transcripts. And you can see, again, that it expands all the way to the inferior frontal gyrus, the insula, maybe some voxels there in the superior temporal sulcus into the gyrus, and the inferior portion of the parietal lobe. Now, sort of a similar pattern when you control just for the number of nouns. So we're not just controlling for naming as a performance score based on the 4 subtasks of the WAB. This is purely for the number of nouns elicited. We get a very similar map, as you can see here. And then there's the other confounder that we were talking about, the idea that you can have apraxia of speech and that can limit your production of a connected discourse that can challenge your everyday speech production needs. And again, the introduction of apraxia of speech from that apraxia of speech task that I mentioned really starts confining the critical voxels for the sentence structure complexity to the inferior frontal gyrus and part of the insula. Now, when we introduce both, the picture becomes even clearer, right? And so just to summarize again, because I think if there's any image that I want you to take home today in your retina and hopefully make it into your brain, is really the fact that when we look at sentence structure complexity as reflected by this principal component analysis, and we control for the ability to name objects and for the level of apraxic speech shown by a patient, it is really a cluster of voxels in the pars triangularis and opercularis of the inferior frontal gyrus and the insula that are the critically associated structures for what we seem to be approaching as syntax. So we are not saying by any means that this is the only way to measure syntax. This is definitely a way to approach syntax, but at least we can say that for some elements of syntax, these are structures that when controlling for the ability to name and the apraxic speech, these are the structures that are critically associated with that behavioral function. So this is the gray matter side of the world. I told you that we were also doing connectome-based lesion-symptom mapping. And again, here on the top, you'll see the connectome-based symptom lesion mapping for that first principal component analysis, looking at the sentence production when controlling for sentence completion, which again is a subtask of the naming domain of the WAB. And just to contrast it, you'll see that there's a network of connections that are more posterior and ventral that support just naming per se. And finally, we said, all right, noun phrase and verb phrase were such strong factors loading into that first principal component analysis. Why don't we focus on those two in an isolated fashion? And an interesting approach to this was saying, what happens in terms of lesion mapping when we analyze what it takes for our brain to produce verb phrases beyond the ability to produce the verbs? So what we did is we did VLSM for verb phrases beyond the number of verbs that patients had elicited, because somehow this probably reflects what is that extra step between just action naming and the ability to construct a more complex structure to convey more complex information. And you'll see that both verb phrases and noun phrases, so verb phrases on the left and noun phrases on the right, both had similar cluster of critical voxels supporting the production of these syntax structures, again, controlling for just the lexical component, which is the number of verbs and the number of nouns elicited. Now, interestingly, the reverse analysis, so looking at the number of verbs, but controlling for the number of verb phrases, did not yield any significant voxels. Same for nouns, controlling for noun phrases. And to me, that's really interesting in that when you think about it intuitively, is there something else that you need to construct, to elicit a name of an object that you wouldn't need in order to build a noun phrase? And intuitively, the answer is not really. And basically, the VLSM is showing sort of that same story, right? That there are no critical voxels that are supporting just the naming per se when you control for the ability to produce a complex structure that involves that name. Does that make sense? And again, we looked at the white matter side of the story, and what you can see here is that there's a very similar pattern, and these are the same. The top row is just based on centroids of those white matter connections, and the bottom row is just ball and stick figures of significant connections that supported. Again, on the left, verb phrase production, controlling for verbs, and on the right, noun phrase production, controlling for nouns. And I think it's pretty remarkable that independently of the ability to, or what the syntax class is, it doesn't matter if it's noun phrases or verb phrases, the white matter structures support their production, that is the production of key elements that are important, the structuring of a complex sentence, both have pretty identical white matter structures. The only, I think, difference was that the noun phrases had this more anterior temporal connection to this frontal structure. But when we think about those results that I showed you from our prior paper, looking at dissociable white matter networks that support the production of nouns separately from the production of verbs, there's something happening that makes noun phrase and verb phrase production converge in terms of the white matter tracks that are needed to support this function, compared to just nouns versus verbs as a naming of action and object function. And again, the reverse analysis did not yield any significant connections. So sort of to summarize, we used NLP to analyze transcripts of connected speech elicited by patients with post-stroke aphasia. To us, this was a very important and challenging endeavor, mostly driven by the fact that we need to increase the ecological validity of how we're measuring speech production in patients. These tasks, the picture description tasks, allow us to resemble more closely real-life language demands, but again, this comes at a cost, which is that they're very challenging to analyze for a human being at large volumes of data. So one limitation that we can point out is that picture description tasks per se are great in eliciting connected speech, but we also need to acknowledge the array of syntactic structures that they can elicit is also primarily constrained to statements employing declarative present tense, right? You're describing what you're seeing. The kid is flying the kite. So an alternative is using, for example, story narration or semi-structured interviews. And I'll show you future lines, but one of the next steps is looking, for example, at Cinderella story, asking the patient to tell a story that has past tense, present tense, and hopefully future tense, and trying to see if a more varied or a more diverse array of syntactic structures can actually help us or shed light on the way that our brain supports syntax and syntactic structuring during speech production. Another limitation was that manually analyzing this amount of speech, as I was saying, would require a lot of training. And we said, all right, the alternative is NLP. It's automatic. It requires little human input once you transcribe and basically prepare the text for input. It is highly reliable, and we've shown that the error rate, even for aphasic speech, is pretty decent. It was about 4% when we're being very conservative with our error rates. So we hope that this will actually open a line of research where we start using NLP to understand language in patients with aphasia. Another limitation that I already highlighted was that this first principal component just mainly captured speech quantity more than complexity of the structures, and so that's why our alternative was using the second PCA. And then the idea that any analysis of speech is constrained or confounded by naming ability and apraxic speech and other variables, and so it was very important for us to use these control variables, these regressors, to try and get a more pure measure of syntax or structuring during speech production. I told you about our bias towards object naming. So as alternatives in the future, there clearly needs to be perhaps a task that measures action naming as a counterpart and see if that affects at all the voxels or the connections that are critical for the syntax variables. And so as take-home message, really sentence production, but in particular sentence structuring and complexity, that second PCA during connected speech seems to rely on the posterior aspect of the inferior frontal gyrus and maybe the insula, and what seems to be a frontoparietal network of white matter tracts that support critically speech production with this increased complexity. So in terms of future directions, again, analyzing the connected speech from other tasks like the Cinderella story, analyzing syntax comprehension. We've shown that there's clearly a bias towards a dorsal structure of the posterior inferior frontal gyrus, which I have to say many functional studies have shown that Broca's area has some has some role in the production of syntactic or syntactically complex structures. Hopefully with lesion mapping, we contribute to that story, saying it may be a critical area for sentence structuring during speech production. Very interestingly and importantly, I think, is now that we've done this huge effort to determine the error rate, and that was done from a human perspective, comparing how the human analysis of speech compares to the automated NLP, because that may give us more confidence in just using it in future studies, as well as comparing the performance of NLP with things like aphasia bank, which are sort of an intersection between an automated component that has a heavy human input in the tagging of the components of the transcripts. Using NLP to analyze lexical features here, which is focused on syntax, but because the parser is so good at tagging every single word, then we can take those words and those tags and see what happens in terms of age of acquisition of the words, imageability, frequency of the words that are being used. It can give us a lot of powerful tools to more closely dissect how the lexical features of speech production may distinguish different types or subtypes of populations. And I think on that note, the idea that there are other confounders that we can use, I think working memory, and when you think about those first VLSM figures that I showed you that spanned into the dorsal prefrontal cortex, which I think makes a lot of sense when you think about just that first principal component analysis being sentence production, right? Do you need a working memory load? I think that's a very interesting question. And then can we use from NLP derived variables, potential biomarkers that may help us not only subclassify types of aphasia beyond the classic Broca's versus Wernicke's, which in clinic we know doesn't always fit so many of our patients. Can we characterize different kinds of patients based, for example, on syntax patterns? And that may translate in more tailored and perhaps more efficient rehabilitation. So I think that's all I have. I, of course, want to thank Leo Moni as my mentor and driver of this research, and John Delgaso at MUSC for working so hard on making this NLP work. Yanina, who had a great role in helping us with the error rates, and then all the folks at USC, as well as Greg Hickok and RJ Hillis, who gave us input into a manuscript that we're writing with these data. I think this truly reflects the spirit of C-STAR, so thank you for having me. Let me know if you have any questions.
Thank you, Zeke. That's great. Do you want to take questions from your own room first?
Sure. If there are any questions here locally, I'll make sure to repeat them for the audience there. I think we're good on this side.
I got a quick question, Zeke. This is Julius.
Hi, Julius.
Hey, so I wanted to ask you, with your second component, what you're identifying there is the basically Broca's area and parts of the insula. What is this area doing? You want to speculate that? And, you know, a couple of more, is this a part of the parser, if you will? And is this necessary for comprehension?
Those are great questions that I don't think we have an answer for yet, especially the second part. I think the second part of your question, the answer will come from looking at syntax comprehension tasks, looking at whether a patient's ability or a participant's ability to distinguish the way syntax translates into, for example, pictures, right? Just showing a task like we've discussed before, where you give them a sentence and you ask them to choose which of the scenarios that you're showing them reflects that sentence. I think that's going to give us an answer to the second part of your question. As to what Broca's and potentially the insula are doing in terms of syntax production, I think when we put together functional studies, electrophysiological studies, and now voxel-based lesion-symptom mapping of syntax, or the things that different studies have tried to attempt to determine as syntax, because the variety of the tasks are so different, I don't know that we can truly say we have a specific story here, but there's clearly something about this posterior inferior frontal gyrus that tends to be involved with the ability to somehow organize speech into an output, right? I think if you're very, very liberal with your thoughts, and you think about Hickok and Puppel's new or more modern approach to thinking about the way language is processed, we keep seeing this more dorsal stream that has to do with some form of production. And what production is, I think, varies a lot based on the tasks that have used to be called syntax. But I think we may get a very good answer when we look at comprehension of syntactic structure and structure complexity, and try to see if there's a dissociation. Because if we see that there is indeed a more ventral support for syntax comprehension, I think that will be very telling.
Hi, Zeke, can you hear me? This is Bree.
Hey, Bree.
Hello. I have a question in terms of how good the NLP parse is going to be if you have someone with particularly empty speech. So someone who says that, there is, that one, there. Like what type of, how good or how much of the error rate is going to change for someone who really has very little content words but has just a lot of hanging utterances and things like that?
Yeah, that's a great question. So one of the things the parser is really good at doing is determining whether a part of the speech is a fragment or an actual valid structure. So if the patient is not using, for example, a main noun or a main verb, the parser is not going to tag it as a verb phrase or a noun phrase. It's just going to say fragment. And so you can measure the number of fragments that are not valid tags and get some sort of poor syntax measure that then you can use, for example, to do a ratio. You can say, number of noun phrases over fragments. And I think that's an approach to go about it. I think Wernicke's patients can be a really good model to test the parser because the way their speech is produced can be really challenging for the parser. In our case, only 3 of the 65 patients had a Wernicke's kind of classic profile. So on the positive side, I don't think it was detrimental to the whole error rate. That 4% with a very liberal approach is, I think, pretty decent. I hope you guys agree on that. But I think if we could rerun this in a larger Wernicke's patient population, we could gain a little more understanding about how the parser performs in those cases.
I have another question, Zeke. Oh, sorry. The step where you do the annotation with the commas and the full stops, I seem to understand that was done manually, right? So even though you had kind of an algorithm, or you had a definition, I should say, criterion, if it's longer than 2 seconds, it's a full stop, but then there was manual correction of that. And I was just wondering, have you tried just going with the criterion and then seeing how much of a difference it makes whether you manually correct these or not? So what I'm getting at is whether it would be possible to do this in an automated way as well.
So the answer is yes, this was humanly done. It was an inhumane task, but it was humanly done.
Hence the question.
Yeah, and I think that the way I imagine the future for this, if you guys allow me to dream for a little bit, is as speech recognition systems become more and more reliable, and as their reliability increases, hopefully their reliability for aphasic speech increases, I think we could potentially get rid of the human part of this, where you just, from a clinician perspective, imagine just having the patient sitting across the desk in your office, you give them the picture description, you ask them to describe this picture, the machine automatically transcribes it for you. It adds the commas and the periods based on the pauses. It sends it through the NLP pipeline, and then it gives you all these measures that somehow allow you to classify your patient into a specific category of subtype of syntax pattern, for example, or lexical pattern. And then you tailor not only diagnosis, but I think more importantly, treatment. So the answer is yes, this was humanly done, and I think it can be done automatically. We just need to get a little, or quite a lot better in terms of speech recognition. I have an accent and I still get Siri asking me, what did I say?
Right. Can you see on your screen the chat box with the questions, or do you want me to read them? You have a couple of questions, I think, on the chat box.
I see a question from Susanna. She says, I hope you can replicate this approach with bilingual aphasics. It would be very interesting to see which language emerges as the more dominant after stroking so-called balanced bilinguals. I think that's a great question. Bilingualism per se is such an interesting concept. I think this is a very encouraging question in that, you know, can we get more subtle things about syntax and structuring of sentences during speech production that allows us to get or gain more information about the patient's language patterns in the brain, for sure.
I think we're good. See, we don't have any more questions here for you.
All right. Well, thank you so much for having me.
Thanks so much for your talk. It was awesome.
Thank you.
Great.
Thank you.
So for the